This bit is going to require more concentration. This bit is not all as intuitive as the previous bit, and we'll be outside, a lot of us, including my comfort zone. And we've got to thank Dr. Anubhav Khan, who's here today, because we would never have managed to do such advanced cutting-edge genetics without him. So I think he deserves a round of applause before we start. So that made you embarrassed, didn't it? Quick one. Everybody knows what DNA is now, don't you? Because we all heard about it during COVID. I hate saying the instruction manual for life, but it is what it is. It's the code which we have in every cell of our body, which tells our bodies how to make proteins, which basically affect what we are. I'm not going to go into detail on this because I remember I told you all about this the last time I spoke to you. So, the quick bit of genetic background. A SNP is a single nucleotide polymorphism, which just basically means a little bit of DNA that codes for something. And changes in those instructions can change both appearance and function in an organism. And okay, I'm really sorry these are not Exmoors, but I just thought it was kind of fun. Underneath these pictures, there's little bits of what the genome is, and the pictures show you how just very small changes in that code can make very big, obvious differences. So I just thought colour was a really nice, obvious one, because we can all see, most of these have got, a similar gene, but a tiny change in it and whack, different colours coming in. So genes influence what we are, they influence what is called our phenotype, which is how we look, how we behave, what we come over as. So you've all read Sue's book, haven't you? So you know the history. At the end of the Second World War, we had this extreme genetic bottleneck. The Society's phrase that they've used very sensibly for many, many years has been, we had a genetic bottleneck of about 50 individuals from which we had to recover. Why we went down to 50 individuals is another story, so read Sue's book. But what you see happens with the genetic bottleneck. You have lots of variation. Each of these little coloured ponies represents a slightly different genetic code in that particular pony, which is why it's a different colour. When you go through a bottleneck, and a lot of them get killed off or disappear or don't breed or whatever happens, only a small fraction of the genetics comes through the bottleneck. So although your population may expand over time, once you pass through the genetic bottleneck, as you'll notice, we're not getting all those colours back. We've just still got the same two colours that made it through the bottleneck. So we don't increase our variation until a long time after the bottleneck. When we start to see mutations, we'll start to change things again. But we're pretty close in genetics terms to our bottleneck. So, what we've got to think about is what variation came through that bottleneck. So, another slide just to show you the same thing. Size of population. So we've got a nice big population, goes through a genetic bottleneck. This is time. And it shows you that you start after a bottleneck with small genetic diversity, and basically you only gradually rebuild it. This one is over time. This is population size. So before the bottleneck, we had a much bigger population. We had the bottleneck, the population crashes. After the crash, down here, one of two things can happen. You manage your population well, you keep an eye on how you're breeding, you make sure you keep your genetic diversity, you manage things sensibly, and you go up to recovery. You don't do that, and you're on the road to extinction. Now, I sincerely hope, and I'm sure it's not true, that anybody in this room wants to see our ponies go extinct. So, we are at a very important point in our society that, for the first time really, and way ahead of many other societies, we can actually combine the pedigree data that we have from our stud book with new molecular methods to really investigate what is the state of our population, and hopefully it will help us see ways forward to make sure we keep our population healthy and grow it. So, another quick bit of background. That's a photograph really from our lab of when we've been doing DNA extractions, and this is just a slide where different individual ponies along here, we were checking that we'd actually got good DNA out that we could then send off for sequencing. So we were removing DNA from the hair roots. There it is. So that's a real slide from our lab. We sent it off for sequencing, and the sequencing comes back with this, the letters of life. I think most of you will have heard of that. ATCG, Gattaca, the film, if anyone's watched that. This is what comes back, and then we interpret from that recipe, code, whatever you want to call it, of those 4 letters, what's going on. So the implications of all this is it can inform within and between breeds relationships, and that leads to better informed conservation. Now, this is what all rare breeds and all management people do. Annabab's worked with tigers a lot. We had someone in our group who was working on rhinoceroses. We were applying the same techniques to these rare animals, the way we get the DNA, the way we look at it. This is how we inform ourselves for the future. This is what we did. We did a pedigree analysis first. We investigated the stud book records, and good on our society for keeping good stud book records, to identify extant founder lines. That means this is work that Sue and Gay did very early on, that Michael then continued and I've continued. We looked at which ponies had survived the bottleneck, so what were right at the start after that crash. And we looked at those. They were the ponies which started, you could only be descended in the current population if your parents, grandparents, whatever, survived that bottleneck. The ones that survived that bottleneck are our founders. So all the ponies in our current population came from those founders. And what we did first was we had a look, who were the founders, which founders are still represented in the current population. This is going to become a very moot point in a little while. So how did we do it? We went out and collected hair samples. After having worked out what we thought all the founder lines were likely to be, we went out and collected hair samples from them. And there are a number of people in this audience, thank you, all of you. I'm not going to name you all because there's a lot of you who very kindly went and made a big effort to collect hair samples from your ponies. And we chased around quite a few too. And we got hair samples that we thought represented a really wide cross-section of the breed, as wide as we thought we could, using the stud book records. And people were absolutely marvellous at this. They made huge efforts, big collective effort. Thank you, all you breeders. Then we did DNA extraction and PCR amplification using these Keogin kits. Now, I'm saying a thank you to Elizabeth Kilbride, our technician here. Elizabeth, although she's younger than me, did this as the last project before she retired, and she stayed on a bit longer than she might have otherwise done in order to help us finish. She was so committed to this project, she took her retirement a little later than she might have done otherwise to help us. And Elizabeth, I know, I bribed her with a lot of chocolate, made a huge effort to get really good, clean DNA samples from our hair roots and make sure everything was really absolutely spot on, so we didn't waste money sequencing samples that weren't perfectly extracted. She's fabulous, and I've got to thank her. So she extracted it for us, and then we sent it off. The little bit of DNA you get out has to be amplified. This is a standard procedure that everybody uses. And then we sent it off to be sequenced. So we sent it in 3 batches to Novagene because we were just making sure our batches worked. We were making sure we've got full samples. And we were also choosing amongst the samples we had which ones were likely to A, give us a full range of what we were looking for, and B, give us a way to read relationships nicely and to look for particular markers on what's happening within the genome. So we were fairly selective on that. So Annabab Khan, Alex Brumwell, one of our students, the lass who's going to get an Exmoor tattoo on her arm, hasn't yet. They did a lot of very clever things with computers doing bioinformatics studies. It's a lot of supercomputer time, isn't it, Annabab? You do a lot of programming, you put in lots and lots of data. We're talking about millions and millions and millions of comparisons done by these computers, but it's not, you could not possibly do it by hand. Believe me, I'd have tried if you could. It's comparing lots and lots of little data markers with each other across all our samples. It took a very long time and a lot of university supercomputer time. And inside of that, we looked at, get used to this symbol, mitochondrial DNA, which I'm sure most of you know is the little circular plasmid which you inherit only from your mum, so it's female lines, and you find it in most of the cells of your body. So that's the mitochondrial DNA is your mum's DNA. It's the way you trace female lines. We did a Y chromosome analysis, which means, you know, that's what makes you male if you have a Y chromosome. Sorry if that's politically incorrect. But yeah, we were looking, yeah, biologically. We were looking for the Y chromosomes. And then Biparental pairwise relatedness and inter-herd means the relationships within the herds that we got samples from. We mostly did this on herds where we had 3 or more samples from a particular herd, and the relationships between the herds, so that we got a picture of how inbreeding was within specific herds, but also across the whole breed and the relationships of each of the herds to each other, which was a way of informing ourselves on basically how the inbreeding's running. We then did inbreeding based on runs of homozygosity. Please don't be frightened by that. You're going to see the letters ROH come up quite a lot. A run of homozygosity is a stretch of DNA, that code that we carry, that is identical by descent, i.e. exactly the same sequence, so the same set of letters, A, T, C, G, or whatever, that you've inherited from each parent. So if you've inherited a run of A, T, G at one particular point on the chromosome from one parent and a row of the same letters from the other parent, that is called a biparental inheritance of run of homozygosity. And it's used as a measure of inbreeding. It means you've inherited exactly the same things from both parents. Then we got some really cool stuff. We got to looking at deleterious alleles. Those are alleles, it's called genetic load in relation to inbreeding. It's looking at alleles, bits of code that could cause disease or could cause problems or may somewhere down the line be less healthy for you as you're functioning than a better one. So we were looking at where possibly problems are happening and the proportions and how these deleterious alleles are inherited according to how inbred you are if you're an Exmoor pony. And then we started, this is why I said hang on with the parasites. Annabelle actually had this really great idea that we were going to look at the relationship between parasite load and inbreeding, because we thought that might be quite relevant too. That was only a very small sample size, but we did it. So here we get onto the genomics results. Lovely foal, isn't it? Still wet after the birth of Bam. We have a lovely film of that we could show you. We ended up with 55 whole genomes, which is actually a lot, isn't it? Yeah, not many people have done 55 from one breed. In fact, I don't think anyone has. So we were very pleased with that. That means the entire genome of each of 55 ponies. We ended up with more than 100 mitochondrial sequences from the females. So that gave us more than 100 samples of just that little circular plasmid of DNA for tracing the female lines. We got, I think it was 12 male Y chromosomes that we put into the analysis. And we ended up with these runs of homozygosity that we could look at, and looking at the way they're distributed, how many there are and where they are, gives us measures of the amount of genetic diversity in the population. Now, if you remember, that bottleneck caused us to lose diversity. And it's very important for us to know how much diversity we've got in the population and where it is in order to preserve it. Just on our stud book alone, we couldn't know that. So that's where this study came in. This is where we combined the modern molecular lab methods with the stud book to see where they were. This is literally just from stud book data. You can see that along the bottom, it's the years, and there's 1980, 2000, 2020. And on this side, it's the number of foals born. So you can see that back in the 70s and 80s, we're ticking along. Then we get into the 2000s and our population goes up. And then about 2010, starts to drop off. There's 2020. And after 2020, the number of foals being registered in the stud book drops off. There's a thing called the effective breeding population, which means basically the amount of possible pairings you can have to increase the diversity within your population beyond which you can't go. The number of stallions and mares limits. If you've got very few stallions and a lot of mares, or the other way around or whatever, it will limit how much variation you can have coming through your population. And you'll see that it kind of follows, not exactly, but kind of follows numbers. And this is a very important measure that's used in a lot of conservation programs, effective breeding population. So there it is looking high when we've got a foal registration. Here it is dropping off. And here it is in our latest figures, almost back down to our trough there. So something's going on And I actually can't tell you what happened here, why this dropped off. I mean, it's possible it's a post-COVID slump. Possible the market's fallen out. I'm not sure, but something's happened and we're getting back down to very low levels again. Now, I'm not saying we necessarily can or should try to aim back up here and preserve it at that. Maybe there isn't the market, maybe there isn't the space, maybe we can't. But it's a bit alarming to see that long-term trend and to see it get so low. So this is something I think the society has to pay attention to. What is happening with our registrations? What is happening with our mares and stallions? How many stallions have we got? How are they being used? To keep an eye on our effective breeding population to maintain it. And this is something that's going to need every year continuous ongoing monitoring. This is actually not too difficult to do. This can be done from registrations data and be followed regularly. I think Michael's done work on this before. Is something that's not too difficult for the society to do, but I think we should be watching this, doing it every year and informing you all so that you actually know how it's going. If it it goes back up a bit again and then restabilizes, I personally won't be too concerned. But if it doesn't, I really want to know what's going on. And I can't tell you why that's happened. So you've all heard us, well, quite a lot of you have been around for a while, have heard us bang on about founder representation. But it's critical and anyone can follow this one. The founders are the ponies that survived the bottleneck. The founders are the parents, grandparents, great-grandparents, however many generations of all the ponies we now have alive. This, again, is done from stud book data. So, this is the founder lines, all the ones that survived the World War. The males are shown in blue. There's always going to be a bit of unevenness in that. The females are shown in green. It doesn't matter which founder line's on there. I could tell you, but I didn't put it on here because it would have made the graph look horrible and you'd never have been able to read it anyway. But what this tells you at a quick glance, some lines really, really well represented, which we're delighted about. Don't get me wrong, this is a really good thing. Some lines are teetering on the brink of extinction. We've got to pay attention to that. Why are some teetering on the brink of extinction? Why are some doing well? Is it because of where they are? Is it because they're not being bred from? There are many, many reasons for that. But it's something we need to pay attention to because it has declined since the bottleneck. There were founders that could have been on there who've left no descendants in the current population. So we have had a continuing erosion, i.e. a continuing bottleneck in our genetic representation since that 45 crash. Some have disappeared. We need to watch that. So currently, worldwide, as far as we know, 28 female founder lines extant, which means 28 of the female founders that made it through that 1945 bottleneck have got descendants alive in the current population. 25 of them are still represented in the UK population. 3 of those founder lines are only represented in populations abroad. That's okay, they're still represented. 13 female lines have been lost since the post-World War II genetic bottleneck. So, we had this bottleneck, it wasn't looking good, and we've lost even more since. We need to pay attention to this. Now, I know you've heard Sue say this for years, Gay say this for years, Michael say this for years, me say this for years, but it's still true. We cannot afford to see more erosion. Pay attention. Uneven representation of founders in a population is a problem. It means that we have a lot that are vulnerable. I'm not saying run out and only breed from the rare ones, because all you'd then get is a seesaw and it'd go the other way. And that's no good either. I'm saying, think about your pedigrees, think about what you're doing and pay attention. There are people in this audience, I can look at some of them now, who I'm very pleased with because they've done this for years and they've done a great job, but we need more people to pay attention to this too. And years ago, I was told all the rare bloodlines were rare for a reason because they're awful ponies. There are people sitting in this audience that have bred Exmoor breed supreme champions from rare bloodlines, which everybody admires. And strangely enough, their bloodlines have picked up tremendously and they're looking a lot better now. So whilst there may be reasons why some are rare, It may be things like bad luck where they are, because they're in non-breeding homes, because they're particularly good at being ridden, something like that. Look at it and decide. Just putting in a plea there, but it doesn't necessarily equate with an actual problem with that line. It can be circumstances. Right, Y chromosome analysis. This is quite exciting. We didn't have that many samples of Y chromosomes, sadly, and for all sorts of reasons, we didn't manage to do as much in-depth study on the Y chromosomes as we would have liked to. We didn't have that many samples and all sorts of things happened. This is something we would like to do more. We would like to sequence more males and do a more thorough look at what's happening. But on this side, diagram A, it compares Exmoor's other domestic breeds. We had 23 of those that you could put in that had comparable levels of genetic information available, which means that we didn't cover all the breeds by any means, because a lot of the breeds have got no genetic availability. Other breeds haven't got good enough quality or comparable to ours, because ours was very well done, that we could use. But with the other breeds, we did the comparison. So the whole, we call it a cloud when things cluster on a graph like this. The cloud of domestic horses is mostly here. The cloud of the Przewalski, you can see there, so the domestic horses are the orange dots. The Przewalski are in blue, and as you can see, the Przewalski's Y chromosomes, there's 2 there and 1 there, so they're a bit spread. Now, one story, which I think is probably true, is that the Przewalski bloodlines, when they rescued them from pretty much extinction, they knew that there was one that was not a purebred, it had some domestic blood in it. So that might explain why 2 of the samples are there, nicely separate on their own, nice and distinct, and 1 is across here in the domestic group. The Exmoors are green, and there they are, clustered in amongst the other domestic horses, with the 1 exception. So there we are, we'll show you our 1 exception and our cluster. Then this side, this is just the domestic horses. Now, where they'll fall on these graphs will depend on the level of focus. If you imagine you're looking through a magnifying glass at something, you look at the beetle on the table and you see the whole beetle. Then you get the magnifying glass out and you have a quick look and you'll just see a bit more colour on the beetle. And then as you've got the magnifying glass even closer, you'll maybe see all the fine scales and things like that. You can think about the way we did this analysis as exactly that. We took a broad brush look, then we took a slightly closer look, then we took a slightly closer look again, and each revealed slightly more detail. So on this one, this is just the domestic breeds. There's our Exmoors. Don't they look nice clustering on their own? I was really pleased when we saw that. So the fact that the Exmoors cluster so beautifully there, away from the others, was a great source of joy to me, and thank you, Annabelle. What I would like to do now is more study into the Exmoors themselves and try and get even finer detail. What you could see is we saw the beetle, so we knew it's beetle. There's our beetle. And then we got a bit closer with our magnifying glass and we're starting to see the colour of our beetle. The next move, if you'll forgive this rather ridiculous analogy, which only just popped into my head, to see the real details on the beetle, we need to do some more. But I would say this is a really nice, promising start. You know, we have started this analysis, we are seeing something different, we are seeing them cluster nicely. Now we need to look some more. We didn't have that many males in our sample, and for various reasons we couldn't go further with this analysis, but that's a future work to do. Go further into it, see what we actually really have got. Because to inform conservation properly, you can't play guessing games. You've got to know what you've got, and this would be a cool thing to do. This one really made me scream. Oh, good God, I danced up and down the corridors when we got this one. This is the mitochondrial haplotype distribution shown by matrilineal founder line. So, to run you through this slowly and steadily, we talked about the founder lines, so we know which females survived the bottleneck. They all have a number. I gave the anchor herd, for example, the number 900, because we can put an anchor symbol into our computer, so randomly it was given the number 900. So anything with 900 means an anchor pony. These, like 900,005, for example, here is Magpie, who was anchor 5 out of their foundation mares. Up here, we've got the 23 foundation ponies, we've got herd 12 foundation ponies, and others. So this is the foundation mares tested by direct female matrilineal descent through their mitochondrial DNA, mapped by their haplotypes, the type of mitochondrial DNA that they're carrying. And as you can see, we've got some variation in that mitochondrial DNA, which is a really good thing because we wouldn't want it all to be the same. So we have haplotype 7, haplotype 9, haplotype 5, 4, 1, 6, 2. I won't go into the details of why they've got those numbers, but just believe me, it's good that we've got those different numbers and we've got some variation. Haplotype 7 is shown twice because actually a lot of the mares came from that haplotype. So this is merely the same thing expanded up to make it easier to read. It's exactly the same thing. They're joined in what is called a network, which shows you their relationships to each other, because each of those dashes across the line means a mutation has happened. There are different ways you could draw this map, but from this map, what it's telling you is that there's that many numbers of mutations between, this haplotype and that haplotype, this haplotype and that haplotype, and so on. The largest haplotype means that those have survived particularly well, so maybe they have better survival traits, better survival characteristics. Yeah, potentially. That's the one where we've got the most in our sampling. So if you come from one female bloodline, you all come from the same great-grandmother, okay? So you should, given that mitochondrial haplotypes only go down female lines, you should have the same one as your grandmother. You can't have a different one. She's not your grandmother if you haven't got the same one as her. So, little interesting one here. Haplotype 6 here mostly comes from that particular haplotype. We found one over here. Oops, not likely to be the case. When we investigated that one, it's because an imbecile hadn't realised that it was a pony with that particular name, number 2, not particular 1, so she put it in as the pedigree of one pony, and it wasn't. It was the other one. It was whatever the pony was called, number 2, instead of whatever the pony was called, number 1. So that was entirely my mistake, and that's completely resolved, and that actually should be up there, and there wasn't a problem with that one. And believe me, I was really delighted to find so few problems. This one, little mistake in the pedigree somewhere down the line. Nothing malevolent, all from the same herd. Mistakes occasionally happen. We're not sweating about that. The really exciting one, which ties into a lot of the historical stuff that Sue has published, is this one. This is haplotype 7, most commonly occurring one in our breed. If you've read Sue's book, which I hope you'll have, again, and she's not paying me to do a plug, by the way, the ponies came through the ones with Thomas Ackland. Yeah, and herd 12, anchor herd, and herd 1 had their origins in the anchor herd. And there they are. All the anchor foundation mares have haplotype 7. Herd 48, one of the herd 48 lines, and a lot of the herd ones. They all come from the same historical origin, which means way, way back in time, their great, however many you want to put in, was the same. This ties in so nicely with the historical story that the anchor herd had, were the foundation herd that Thomas Ackland protected, and the other herds took stock from them and came out. You're going to get variation and mutation, that's fine. I'm not rubbishing any of the others, not by any means, but it's just such a good tie-in that this shows that that history was basically right. And something that I was so proud of, really, really, was, yeah, one of those mistakes was mine, and the other one was just a little bit of confusion. It came out so well. I really, really am full of admiration for all the hard work people have done to keep this right. It's so good. It came out better than I thought it would. So great work. Our mare lines, we know now where they are. We know who's in them. We know our haplotypes. And something that I want you to consider, and I haven't got an answer to this because I haven't decided for myself yet. I'm starting to think about conservation of the founder lines in terms of founder mares, all these lovely ones that we've marked by different colours around on here, but also on the foundation haplotypes, because that's going to be really important too. And I haven't got an answer to that, but it's something I'm puzzling in my own head. Maybe we need to think about how to maintain our other haplotypes as well. So there's a thought for you. But this was such good news. So we've got to be very proud. And as I say, historically, this ties in with the whole Exmoor history beautifully. Then the next thing we did. biparental relatedness and interherd and intraherd relatedness. Here we've got the same story, that there's very little variation between the herds that came from the same origin, which was Thomas Ackland, and a little variation with the ones that maybe weren't quite from the same origin. Not meaning they're not Exmoors, just slightly different origin. And this is showing the relationship between the herds where we had more than 3 samples from one particular herd, just looking again at their relatedness. And the inbreeding across the herds that we tested, there's no significant differences, which is good news. Right, this is the one where, hold on to your hat, we're going to have to think a little bit. I had to. This is the inbreeding based on runs of homozygosity. So I've told you what a run of homozygosity is. It's DNA identical by descent from both your parents. So it's a way of looking at inbreeding. Now, what happens over time, each generation, those runs of homozygous coding get broken up over time. So 10 megabyte stretches, meaning quite big stretch, is from 5 generations ago? 10 generations ago, yeah, 5. Then you can also look at the picture with 5 megabytes, a bit shorter, and that's more generations ago, 10. Yeah, because they're broken up and the runs have got shorter. And then you can look at 100 kilobyte pieces, which have got smaller again because it's from further back, 50 generations ago. Yeah. So it's getting shorter over time. So the very short runs are the ancient ones, but in time, more recent ones are longer because basically they haven't had time to break up. So your most recent one, your 10 megabyte, is from your more recent generations. The 5 is from a bit longer ago. The 1,000 kilobyte is from much longer ago because these things change, get broken up each time you get mating. It alters. The results showed more or less the same pattern across the 50, the 10 and the 5. This one's from the 10. The Exmoors are in green compared with 23 other breeds. And you can actually see, and it was statistically significant, we did a lot of statistical testing. The Exmoors, this is the measure of those runs of homozygosity, which is basically a measure of inbreeding. And this is all our breeds. There's the Exmoor, significantly more inbred than the others. And it had the same pattern over all the ages. So a long time ago, they were significantly more inbred. As it got more recent, they still stayed significantly more inbred than the others, and they still are. But it's called a box and whisker plot. The whiskers are quite long, which means there is actually quite a lot of variation within the breed. So, we've got this very high inbreeding, but we've also got down much lower. So there's a mixture across our breed pattern. The other breeds are lower. Has our population always been discrete? Has it not had other stuff mixed in? Is this an indication of it? Possibly. Some of the breeds, for example, that we included, and we could only include breeds that there were suitable published genomes on that were of comparable quality to ours. So there were lots of breeds we couldn't do, like there's no Fell in there, there's no Highland in there because the genomes weren't available. An example, for example, there is the Haflinger. It's considerably lower than us. I did a lot of research on this, and the Haflinger breed was created in the 19th century by creating crossing an Arab stallion with these alpine mares, which was a great thing to do, but it means their population is more mixed. And therefore, the inbreeding is going to be lower. Our inbreeding stayed high all the way through the history. An interesting thought that, we didn't have major changes in it because they've stayed inbred and discrete, maybe. We don't have answers to everything, but we're working on it. So there you go. Regions of the chromosome, runs of homozygosity, regions of the chromosomes where identical by descent haplotypes are inherited from each parent. An inbreeding measure. Now, these deleterious alleles, these ones that can cause problems. These are the loss of function alleles. These are mutations that could cause disease or serious problems in your animal. We've all got them. Every single one of us, me, you, everyone in this room is carrying a deleterious allele load. We have got alleles in us somewhere that could all cause us to have a disease, but they're not necessarily expressed. You can have one copy from one parent and one from another, and it won't show. thinking about things like haemophilia. You've got to inherit it from both parents to actually show it. These are the loss of function ones, which means if you have inherited the same copy from both your parents, you've got a disease problem, or it could be quite a problem. We put the proportion of those on that axis, and we put the increasing inbreeding on this axis. So the more inbred you are, the further towards this side you are, the more of these potentially bad stretches of DNA you've got, you're higher up here. The colour coding is to show which maternal mitochondrial DNA haplotype you've got. But the take-home message, don't panic about this graph. What it's telling you is, the more inbred you are, you see the way the line is dipping? The line, that tells you the more inbred you are, the more likely you are at some point in the past, your ancestors were the ones who, you lost those genes. So you've been purging them. It's called purging. It's a reduction in the number of bad alleles over time with increased inbreeding. So some of our ponies have been doing quite well. They may be inbred, but they've been getting rid of those alleles that you don't really want to have over time. Next one. This is a similar pattern with the missense alleles. Now, missense alleles are not good in the sense that they're not coding exactly how you want. They're a change from the standard healthy one, but they're not the immediately problematic. They're less serious. They're not so immediately problematic disease-causing ones. And we see the same pattern. You're more likely to have a few more of them if you're not inbred, and you're probably starting to lose them if you are inbred. So inbreeding, and James Speed wrote about this, can cause the bad alleles to disappear because they're selected against, i.e., if you inherited two bad copies, you probably didn't survive to have babies that went on to the next generation. Is everybody okay with that? So it was less significant. The trend is less obvious with the less serious problem, but it is there. So over time, inbreeding is getting rid of some of the bad alleles. It's having a slightly different effect on some bloodlines are shedding them quicker than others, but the trends are there. More work to do on that to actually identify which ones, exactly where they are and the rates they're going at. So this is a work in progress. We're not there, but we have seen significant trends. And I got really excited about this as well. And no, I can't tell you whether your pony's got them or not or whatever, because we haven't got that far yet. But what we are seeing is really good signs. The negative stuff is getting purged out of the population. Not an easy concept, but it's worth having. Parasites. Right, just for fun, because I'm a complete sucker for punishment, I did do a study, which I haven't actually put all the data up here yet because it was too much and we could have been here till 4 o'clock this afternoon and all died. I did do a study which looked at parasite loads related to body condition, and it did show that some ponies tolerate high parasite loads better than others, and it did show that on the whole, our ponies, which are kept very much as a wild population, and I do not worm them unless there's something really dramatic going on, and they live on hill ground, tended, compared to other studies that were published, to have a lower parasite load overall. For example, the Sable Island ponies and other feral populations tend to carry a higher load than the ponies I studied. But it was a very small study. It was very limited because I could only do scorid, and this is another one that needs further investigation. Do they actually have a better resistance to parasites than other breeds? I don't know, I can't say that. But it was an interesting little hint that they, on the whole, tended to have quite low parasite loads. But when we looked at this, what we saw was, on this side, the higher up here you are, the more fecal eggs per gram you've got. So it's 100, 200, 300. So that's increasing, that way is increasing parasite load. And this way, we're back to those runs of homozygosity. So the further you fall on the line here, the more inbred you are. And the dots are individual ponies. And again, this isn't a big enough sample to say this is definitively true, we can really prove it. But it was a small sample and it was worth doing, and we did a statistical test on it. And it gave us a hint that one thing that did tend to happen was the more inbred you are in this particular group, the more likely you were to have a high parasite load, except that one. And one with the highest parasite, the one with the highest inbreeding, we got a Ziltron all the time. Yeah, we did this every year. She always had nothing. So I haven't gotten, again, in a way, this study is raising a lot of really exciting questions which I can't answer. I don't know why that happened. But that trend is significantly statistically significant, but it is on a small sample. And we have got one that, bucked the whole trend. So this is, again, something I think it might be worth investigating and looking into further. An interesting thought, I felt. So this is something that we need as a society to take really, really seriously. Continuous monitoring of the genetic health and the environmental adaptability is essential for maintaining a viable Exmoor pony population. If we don't do this and we don't keep doing it, we could get a sudden change that we're not aware of and have a horrible surprise jump and bite us before it's, and it's too late. Sue, Michael, Gay, me have all been doing some of this monitoring, but this is the first time we've been able to put the genetics evidence in with it as well, which makes it considerably more robust, i.e. that means we can rely on it more. If we don't keep doing this, as you saw with the effective breeding population, as you saw with these other trends, we don't know where the trends are going. So what I'm saying is, this is great, we've got these results, but we have to keep doing it. Otherwise, something could happen, we see a change, we don't see a change coming, and we suddenly go, what happened? A thought for you, Fell-Fold syndrome. Fell-Fold syndrome was a mutation that they didn't see coming and went through the breed because they weren't being able to monitor all the time. And you all know what problems that caused. I am full of admiration for the way they got past it, but that could have wiped the whole breed out. And they were lucky they spotted it before they had got to absolute crunch. We need to keep watching to make sure nothing happens with ours, because it could. I'm not being scaremongering. I'm sincerely hoping that everything's great. But you can't guarantee this, and you need to watch. Right. This is another thing that we're very, very excited about. We all go, the Exmoor Pony, the original off-roader Exmoor Pony, ancient origins, Exmoor ponies look like cave paintings. The speed papers did that measuring of ancient bones and ancient skulls against modern Exmoor ponies, and lots of similarities. This is a really exciting thing. We've always said our breed is an ancient breed, but can we prove it? Not at the moment. But our genetic analysis, thank you, Annabab, this is a thing called an admixture graph. That means that you compare the genomes of our ponies against other published genomes to see where they fall in a tree, starting with ancient animals through to modern ones. This is, again, something you're going to have to think about. That is, when it's in grey, it's called a ghost population. That means it's a population that we know from the genetic markers existed, but doesn't exist at the moment, or at the very least, we don't have samples from, but probably doesn't exist at the moment. So it's something that they were descended from, that became extinct. So it started with one of the very old ancestral horses that was like the ancestor to modern donkeys and horses and everything. The arrows don't go entirely on the length, it's the numbers next to them, but that kind of doesn't matter in this bit. The arrows show that from that ancestral proto-horse, we'll call it the proto-horse population, it split, and you've got the donkey coming off here, and you've got a ghost population of horses that no longer exists probably at the moment. A change happened with that, and this branch went off and became the Przewalski. Another branch came off down here and formed another ghost population. That ghost population split, and on this side, 3 mutations to another ghost mutation population, 5 mutations, and it became the modern Icelandic horse. That ghost population, 18 mutations, and it became the Shetland pony, as we see it today. But the cool bit, 6 mutations to a ghost population here, 1 mutation, our Exmoor samples. That we can say is that they are closer, mutationally wise, to these ghost populations of early horses. They come off the tree earlier, as far as we can see at the moment. We can't definitively stand up and go, they are the ancestral horse, they're the oldest living thing. We can't, but we can say we've started to unearth evidence that makes this look quite a good possibility. We know from records that the Icelandic horse was isolated for 1,000 years plus.
Yeah, 971.
There we go, 971, the Icelandic horse was separated, and they're a little bit further away from that ancestor than we are. Shetland ponies again isolated on Shetland. And then we've got the arrows saying modern breeds, because the other breeds that we could study, and like I've said previously, we couldn't include everything. There weren't genomes available for everything. It couldn't be done. But all the other breeds we looked at come down, some from a ghost population that split off from the same ghost population the Exmoors did, some from the Icelandic horses, some from the Exmoors, some from the Shetlands. And the other moderns that we looked at are here, and are various mixtures of all this. So when you think back to Schaefer, who we talked about in the first part, Michael Schaefer talked about migration of the early horses, because the very earliest horses, which look like little foxy things, started out in the central plains of North America. And back then, when North America was joined to Asia, between stages of glaciation, waves of horses migrated across the Bering Straits land bridge, came into Eurasia, and then separated off and became different ancestral populations. You know, they became donkeys, they became Przewalskis, they became modern horses. And within the modern horses, which is from here downwards, they split off into those 4 primitive types. And there's your Exmoor. And Schaefer said that morphologically, and in his beliefs with their behaviour and how they looked and what they were, he believed that they were one of the closest things to that primeval pony, you know, that ancient ancestor that got all the way across and hit the Atlantic Ocean basically and couldn't go any further. And he said he felt they were the nearest to this. We are not there, but we're getting genetically closer to saying, yeah. We can't say it because we haven't done enough work, but it's certainly looking really promising. If we could show that, which we can't yet, and I'm not saying it, but if that changes the whole conservation status of the Exmoor. They're not just a rare breed, they're a primitive archetype. We can't say that yet, but we can say this is really exciting and deserves more investigation. At the point where they are no longer a breed and they're a primitive archetype, that's it. You know, they're up there with the rhinos and everything else. I am not saying that's what they are. I am not saying that we could guarantee that's what we would do if we did more work. But what I am saying is it's worth pursuing to see. We would need to do more other breeds to look at the relationships between them. We would need to do ancient DNA and then do lots of checking and analysis and see where we were going with this. But it could be done. So we're putting in this plea that Exmoors could be an archetype. We could get very excited about this, but at the same time, their population is not secure because we're watching all the genetic trends and we need to be watching that. And in the first half, I was saying to you, there's lots and lots of literature out there in the rewilding world, but it looks at when climate was most stable across the Northern Hemisphere, and that was during the Pleistocene era. And there are people researching this a great deal, particularly in Copenhagen, but in other places, looking at the mega herbivores, that means, you know, the big animals that were grazing, and saying they are the most suitable things for rewilding in the sense that they interact best with the vegetation, which I hope we've shown you, and that they have the survival characteristics, that make them good for it. This is going to be a growing market. The rewilding market is going to be big. If, as I stated earlier, we want to see our Exmoors still around in 100 years' time, we've not only got to manage their genetics, we've got to manage their environment as well, combined. And one of the ways we do that is by finding a role for them, and the role for them could be in rewilding, ecological restoration, whatever you want to call it. Exmoors are already used for this, but there's a lot of work to do on them. So, what makes a rewilded pony? Ability to be independent, adaptations for survival. Well, we've all seen this. You know, we know the little devils are independent. They do really, really well with really minimal help. So this is cool. We haven't got the genetic base of it yet, but you know, our observations, we all know you put them out on the moor and they do fine the vast majority of the time. Thank you very much. Economic viability, meaning minimal management. Again, you know, we all know, well, I hope we all know, we don't need the vet out as much. We don't have to do as much with them. The wild herds live wild. I mean, my own herd, which has been on Squirrel since 1970 and I've been with since 1982, you know, the vast majority of the time they're doing great without our input. And I'm sure that's true of all the other herds as well. You know, we manage them, we be as sensible as we can, but we don't have to be out there every day going, oh my goodness, you know, let's change its rug, let's do this. In a domestic situation, yes, but in the wild situations, no. Genetic management for population security, guarantee of supply. This growing market for them, because we've shown that they're great for the biodiversity, where are the ponies coming from? I know that people are saying, well, you know, we've seen a decline in the number of foals we're registering. We've got to actually manage the population and manage the population well, not go out and double the number you breed, but make sure we're breeding ponies that are genetically healthy and will still be suitable for going out and doing this job. It is a numbers game, but not principally. Principally, it's about breeding them correctly so we have a genetically healthy population and breeding them that they keep those survival adaptations. Natural behaviour. Okay, you know, natural behaviour means, can they do well in the wild? Can they survive? Do they know how to seek shelter? Do they know how to do things? Contributor to natural climate solutions. Well, we've been talking about that, you know, properly managed grazing, carbon storage. Don't forget it, it's quite a big deal. And that's going to become a bigger and bigger deal. Biodiversity improver. Talking about that. Contributor to carbon sequestration. Well, we've covered that, you know, the subsoil storage. I said to Richard as I was finishing the PhD, this is a really great story that they're helping the carbon storage and the natural climate solutions, and the evidence is starting to look good. But do you think it actually matters? And Richard's a big name climate scientist who talks to lots of groups. And he says, yes, it does. It may not be the biggest one, but it is important. And it's one that we can do. And it also improves biodiversity at the same time. So he said, because I said to him, tell me honestly, if you think this is just a rubbish line to chase, tell me honestly so I don't waste my energy on it. And he said, no, you're not wasting your energy. Do it. And believe me, in our family, we would have said, get lost, wouldn't we, Phil? Best proxy for the ancient wild horse. Well, I've kind of covered that one. You know, minimal artificial selection in population history. Well, there we saw, you know, our population looking quite discreet to the others and hanging in there. Can't say that yet, but, you know, good. Thank you. So, what qualifications have been established for the Exmoor? Okay, economic viability. Well, we know they do well out. Independent survival, just giving you a nice story on that. Natural behaviour, biodiversity improver. Hope you liked my first part. Population security. We need to work on that one. What do we need to do further work on? Contributor to natural climate solutions. Well, as David and other people have kindly volunteered to go out and do sampling and measuring, they're going to be going out and doing that sort of thing. And I will be recruiting more of you, and we will do more investigation of that. We will be investigating the plant diversity, and we will be doing more investigation to continue to add evidence to the evidence. I want it to be that we have built up such a good database with such solid evidence that people will go, well, obviously it's got to be an Exmoor because they've proved it. Contributor to carbon sequestration, same thing. We carry on with the research. We measure this. Richard says this is significant. We're going to go and do it. We're going to say, yeah, this is happening. keep it going. Best proxy for the ancient wild horse? Well, again, we've got all these lovely hints. We've got these things pointing in that direction. I can't stand up and say it yet. I really wish I could, but I can't. It would be overstating it, and I'd probably have Annabab beaten to death for overstating it. But wouldn't it be cool? So here we go. This is the further research we need. The natural behaviour, we're well on the way with. It's a tick and a question mark because we're seeing good evidence it's there. But, you know, some more behavioural studies would be cool. And it's exactly the sort of thing we could recruit master's students to do and other people to do. Biodiversity improver, well, I've already said, you know, we've made a start on that, and I'm going to be plaguing life out of people and sending Vivian out in the midges and things like that. We're going to keep working on that one. Population security, well, you've seen we've done the pedigree analysis and we've done some super genetics work that backs it up, but we're not going to stop if we can help it. You know, we need, this is not something you can sit on your laurels about. Contribute to the climate solutions. Yeah, we haven't really got a measure of that yet, but we'll work on it. There are ways to do this. I've got ideas. We will get on with that. Best proxy for the ancient wild horse is tied in with all of the rest. And contributor to carbon sequestration, same thing. We'll look into it.
The coming generations will have good reason to call us unfaithful stewards if, when we're gone, there are no little horses left on the Exmoor hills.
So that's Mary Everington in 1947. Your scientific advisory panel, we've been having agony for a few days, haven't we, all of us? And we want to say this to you. The coming generations will call us unfaithful stewards if, when we are gone, there are no Exmoor ponies, because we failed to seize the opportunity to safeguard the population and to allow them to recolonise rewilded places across the UK and Europe, thereby ensuring the future population of the breed. Now, I personally feel I could cry again. I feel really passionate about that, and I am not the only one. Anyone in this room is passionate about the future of the ponies. You wouldn't be here if you weren't. So we've got to seize this. We've got to do it. We have to monitor our population. We've got all these opportunities. There is that potential big market coming ahead. We've got to work on it. Our registered Exmoor ponies, we're starting to show what they're doing. We keep doing it. We build the population. We make the population secure. We show their integration into this growing market. We put them into it. Oh dear. Just while I'm got the sword, isn't it? We're totally serious about this. We've really thought about this. We're totally passionate, and we know you are too. So, at the moment, the EPS has been ridiculously bold. I've been to lots of conferences, lots of places. We do a lot of networking, don't we, Annabab, with other academics and other people in other academic disciplines. And we tell them all about what we're doing. And we get, oh, good God, that's so brave that you're doing that. Oh, wow, a tiny little society that we didn't know about is doing that. And we've been out and doing it, and people are really impressed. But we can't just stop. If we just stop now, it could all go to pieces in an unforeseen way. I'll shut up.